Today, we're going to hear about water health in humans, leveraging community partnerships to protect Maine waterways. And we have two speakers. I'm going to start with Robin Sleith first. So Robin came to us. He had his BS, Bachelor of Science in Vermont, PhD in plant science from the City University of New York. And he is not an oceanographer. His expertise is in plants. And this is one of the unique things I think about Bigelow. We don't want a bunch of oceanographers. We want scientists from lots of different disciplines that will come and look at science issues and problems because they bring a completely different perspective. And Robin is a great example of that. He came to Bigelow as a postdoc in 2021 with Pete Countway, who is the inviter of Dr. Shaw. So Pete does a lot of great things for Bigelow. And we promoted him last year to research scientist, which is really a tough thing to go up for a job where you already work, but he did, and he knocked it out of the park, and we're thrilled to have him. Then Rachel Sipler, who I have known for many, many years. I met her as a graduate student, and within about the first 20 minutes, I invited her onto a research cruise to the Gulf of Mexico, where her advisor said, it's on you to do it. So she rented a U-Haul herself, drove from New Jersey to the Gulf of Maine in Florida to go on the cruise, and I've been a big fan ever since. So she has came here a few years ago. When did you 4 years? I didn't write down. 4 years from the Memorial University in Canada, where she was a Canada chair, which is a very prestigious honor up in Canada. And we recruited her to come to start the Water Health in Humans initiative, which is now a center here. And with that, I will turn the microphone over to Rachel.
I want to start with an opportunity to tell you about what we're going to talk about tonight. One is I'm going to talk about some of the challenges that we face here in Maine, as well as what we're trying to do about it, what Bigelow and the scientists here and my wonderful colleagues are putting together to make a difference and to make it a better world for us and a better community. The first thing I want to ask you is, what memories do you carry that connect you to the waters of Maine? Is it swimming in a lake? Is it boating on the water? Is it sitting on a beach? There are so many activities. Is it that one weird thing your friend did when you were kind of on vacation and then they had that conversation or whatever? But there's always a connection that you have, and you connect with the water. It's a memory that takes you back. And all of those memories are connected to the quality of life here in Maine, and that depends on the health of the waterways. So what makes a healthy aquatic ecosystem? And I am saying aquatic because our name is Bigelow Laboratory for Ocean Science. But you know what? Just add water, right? We do freshwater, marine. It's really, if it has kind of some aqueous relationship, we're in. There's a lot of skill sets, a lot of opportunities, and a lot of interest. So I'm going to be talking a bit about both today. Robin's going to focus for his talk on freshwater ecosystems and drinking water lakes. But combined, a healthy ecosystem is a healthy ecosystem. We need clean water, whether it's marine or freshwater, diverse life, productive food webs. That means all the way from the minuscule microscopic organisms that form the base of the food web, all the way up to our predators. We need them all to have a balanced ecosystem. Healthy shorelines that are resilient to change. So if we have these things like we've had storms or we have a disruption in the ecosystem, can it rebound, right? It's kind of like human health. If you fall down, can you get back up? That is a sign of resilience in people and a sign of resilience in our ecosystems. The other thing is, do you have strong community stewardship? Are there groups of people working together, community members, scientists, managers, who are dedicated to making sure that that system stays healthy for the next generation? So today I want to talk about some signs of change, but also some hope for the future. We definitely see early signs of change, but we also see signs of resilience. And where there is opportunity, we're going to help that resilience to be present. So what's pressuring our waters? Warming, storm severity, changing water chemistry, changing land use, pollutants, and harmful algal blooms. So starting with warming, the Gulf of Maine, as many of you have heard before, is warming 99% faster than the global ocean. And as a result, we're seeing Maine's coastlines warming faster than the rest of our state. With increasing storm severity, we're seeing high winds and tidal surges. Just within the last few winters, you've seen these major events kind of back to back, having huge washing events and stormwater inundation. But with that, we're also seeing more storms that have a lot more rain happening in a short amount of time. These big storms, there was one the other night. It kind of came through really quick, it dumped a lot of rain, and then it disappeared. Those big storm events, they carry a lot of water, they move a lot of water, but without the ability for that water to sink into the ground, it causes something we call runoff, but that movement of water across the land. And unfortunately, it's not just the water that falls as rain, it gathers things as it travels down that path. And where it lands matters, because how land is used impacts the pollutants released and the environmental impacts. So these are two kind of schematics. One's an urban setting where you can see that as rain falls and runs across roads, you clearly would get things like petroleum products or gasolines, oils that come off. You see that in slicks. If you ever walked across a parking lot after a rain, you tiptoe really lightly, right? You don't want that in your shoes. You see that color, you see that shine, and you know that there's pollution being carried with it. Another one, which we call point sources, or things that we've identified as major contributors, we often say that farming is one of them, because we know that a farm is a designated area, and we put fertilizers, pesticides, and herbicides on those lands. We also do that in our backyards, which I'll get to in a minute. Don't blame the farmers for everything. But one thing about fertilizers is they've been really cheap for a very long time. And because of that, we often use mixed fertilizers. You go to the store, you go to Lowe's or Home Depot or any store, and you get a bag of fertilizer, and it comes pre-mixed with all of these options. And maybe it's for tomatoes, or maybe it's for shrubs, but they have nitrogen and phosphorus and potassium, and it's all ready to go. But sometimes your soil only needs nitrogen or only needs phosphorus. So what happens to the rest of it, right? We think it's just a bonus, but that bonus runs off the land and into our waters and has a negative impact. Because everything that runs off carries these nutrients and pollutants, good or bad. Nutrients, too much of a good thing. We need it for growth, but if you have too much, just like cake, one slice is good, a whole cake's not great. If you give too much of that opportunity, then the ecosystem can't handle it. It's not balanced for that. So all of this is determined by watersheds. And watershed is where runoff goes. It's an area of land where precipitation, or the rain, snow, comes into the system, and it drains down and collects in a common water body. So a watershed is of all sizes. It can be a puddle, if that's the reservoir you're talking about. It can be a lake, it can be a river, it can be an ocean. But everything is part of a watershed. We have watersheds, for example, across the state. Each major river has its own watershed, which means that as it hits the mountaintop, it's the area that drains into a collected point. If you live here in the Booth Bay region, these are two watersheds of our local water district, Adams Pond and Knickerbocker. So you may live or reside within these potential watersheds, or maybe you live outside of that and it goes directly into the ocean. But everyone lives, works, and moves throughout these watersheds on a daily basis. So things like small actions can have big impacts. For example, if you're just watering your flowers with some fertilizer or some Miracle-Gro, maybe that runs off and it eventually makes it to a water body, a ditch, a river. A ditch isn't the end result, right? That eventually moves and can get caught in the soils for now, but it means that it could be mobile later. Maybe you're washing your car and it's just kind of draining away. All of those suds from the soaps and the wax that you're using, eventually that can make it to our waters as well. Whatever debris gets trapped on your car, that can move. Maybe your septic system isn't quite right. There's a little kind of circle of septic that's showing through. Maybe it's not functioning at its peak capacity. All of these relatively small things that you don't really think about on a daily basis, like, oh, I'll get to that. I need to fix that later. On a grand scale, with everyone living in these communities, in these watersheds, they add up. So small actions can have big impact in a positive way, right? We can fix these small things, think about them, and move forward. So the other thing that we were talking about was harmful algal blooms. As we have these nutrients that are running off from our yards, from our flower beds, they can get into our waterways, and they create something called harmful algal blooms, which are algae or cyanobacteria that negatively impact human or environmental health. Now, there are things called nuisance blooms, right? These are when the water isn't quite as pretty as you'd like it, as clear and pristine as you like it, but it's not really hurting you, right? It's like a neighbor parking their car in front of your house. Maybe you don't want it there, but it's not hurting anybody. A harmful bloom is when the water clarity becomes less than it is optimal and it starts to kill the species that live below it. Or maybe they produce a toxin that can actually kill or negatively impact the organisms that live there or people who are around the water. That's more like your neighbor using their car to run over your foot. There's a more direct and immediate impact. So a nuisance is something you'd like to avoid. A harm is something that we really have to prevent. So some of the things that harmful algal blooms do is they contribute to water discoloration. Again, those green or brown or red, harmful algal blooms come in every color. And if you view it through an oceanographer's eyes, and yes, I am an oceanographer, it's the most beautiful spectrum of colors, right? It comes in every shape, size, and from every group of algae. It's just an array of opportunity for color and growth. But sometimes these organisms can overgrow, causing beach closures, adverse human health effects that can be through some, as my daughter calls it, tummy trouble from eating the wrong thing or drinking the water, to respiratory distress. It can cause shellfish, fish, marine mammal, and seabird kills. And at peak bloom, harmful algal blooms can kill up to 100 tons of fish per day. So a major ecological impact. The other thing is they can be really expensive. On a grand scale, it can be $1.2 billion annually globally for harmful algal blooms, and 25% of that is spent on cleaning up drinking water from these harmful algal blooms. So what do you do about it? We listed all of these different components that are affecting humans in different ways and that we can do to help keep our environment clean, right? So they affect us, but also we affect it. So we developed the Water Health and Humans Initiative here at Bigelow because we wanted to do science with and for our communities. We wanted to put our science into action and in the hands of the people who need it and use it most. So how do we do that? This is an interdisciplinary, place-based approach. There are a lot of recommendations on how to deal with environmental change issue that maybe are broad and you can apply them anywhere in the world. But place-based is different. It really offers a view of the uniqueness of each ecosystem, each community's needs, each opportunity to make a really sustainable approach to fixing these problems, something that we can keep going for generations to come. So it starts with observation, working in and with communities to identify issues that impacts Maine's water health. Then we work on innovation. We develop strategies or engineer products to mitigate these problems, working hand in hand with those who need it. And then we put this work into action. We apply those innovations to strengthen Maine's economy, ecosystems, and coastal communities. For these strategies to be effective, we need solutions that are both collaborative and sustainable. And I don't know about you, but for a number of years, I felt like there were a couple camps. If you wanted to really care about the environment, you couldn't drive a car, right? You had to do certain things. There's all these restrictive opportunities that really felt, when I was growing up, impossible to me, right? I wanted to go out and have a good time. I wanted to see my friends and drive to the beach, right? I wanted to do these things that were, from an environmental standpoint, would have a negative impact. From an economic impact, if you wanted to support the economy, then industries were supposed to be promoted, independent of what the negative impact was on the environment. These holistic views of one over the other don't work. We need to have a complete view of the social, environmental, and economic impact so that we have a sustainable solution, something that we can keep going for years that doesn't negatively impact any one of these sectors. We need people to be able to do the activity we're suggesting they should do. It should make we should be able to see the benefit and want to engage in it. It needs to be bearable, it needs to be equitable, it needs to be viable to be sustainable. So now I'm going to reintroduce Robin Sleith, and he's going to talk to you about one specific project that we're working on here with the Water Health and Humans Initiative, and Robin's continued involvement in working with community and his desire to continue this collaboration moving forward.
Awesome. Thank you, Rachel. And again, thank you everybody for coming out this evening. So I want to tell you a story about one of these collaborations that I think really demonstrates the type of work that Water Health and Humans is undertaking and the value of these community partnerships. And so this story begins on Damariscotta Lake. So this is the Damariscotta River estuary just out the window here. If you go all the way up the estuary, you'll find Great Salt Bay. And if you go a little bit further than that, you might encounter the fish ladder, which hopefully everyone's had a chance to see that in May when the alewives are running. The alewives are going up the fish ladder into Damariscotta Lake. So Damariscotta Lake is the headwaters of the Damariscotta River estuary. And what's going on in Damariscotta Lake has major implications for what's going on in the river and in our coastal ecosystems because of all of that connectivity that I think is really exemplified by the alewives. And so in 2020, folks noticed, this was before I was at Bigelow, but they noticed this green scum along the shorelines in Damariscotta Lake that they had not been seeing previously. And so Pete Countway, who's been mentioned previously, the senior research scientist that I then started a postdoc with, went out, collected this sample and took a look at it and identified it as cyanobacteria. And that's sort of the beginning of this story. But before I talk about cyanobacteria in what might be somewhat of a negative light, I really have to give them their due credit because cyanobacteria have been around for 3.5 billion years. They've played an incredibly important role in our planet as we know it through that endosymbiotic event where you had a photosynthetic bacteria that became the organelle, the chloroplast that led to all of the land plant diversity and production of oxygen that we appreciate as humans today. So those are the good things about cyanobacteria, and the planet would not work without them, but there are also some scenarios where they can cause a good deal of trouble for us. And so those are through things that Rachel already mentioned for harmful algal blooms in general, but specifically for cyanobacteria, there are two main ways that they could be a problem. Just through overgrowth, which is going to lead to problems underneath that. You can imagine there's not a lot of sunlight getting through water that's got covered with cyanobacteria. And then through direct toxin. So that could impact humans, it could impact wildlife or pets. But these events don't typically happen in a vacuum. So as Rachel mentioned, there are a lot of activities that occur that can fuel these blooms. And so that's a really active area of research, is understanding when you have something like this, when you have a change in the landscape or the change in the ecosystem, one of the really important questions that we want to get to is, well, what's going on here? Can we find some solution that can point us towards some type of restoration action that we could take? And so that's where this collaborative work kind of began. And so we were actually approached by the Horizon Foundation to develop a collaborative proposal with Bigelow, bringing the science, and then Midcoast bringing their stewardship and the community around Damariscotta Lake to work together to understand these cyanobacteria issues. And so I have to shout out Patricia Neese here, who started at Midcoast around-ish the same time that I was starting at Bigelow, and has been my partner in crime for this journey, and just a great person to have at a local conservation organization. So Midcoast Conservancy does a lot of great work throughout the Damariscotta River watershed and beyond, over towards the Sheepscot, over towards the Madomic. And so Patricia is just one piece of that organization, but lucky for me, came to that organization with a limnology, so a deep understanding of lake science. And so that allowed us to really just kind of get to work on this project. And so one of the reasons that I am sharing this story with you today is that this project has truly been kind of about collaboration and working together every step of the way. So we're out on the lake. This project has been a 3-year project. We're in our final summer. now, and we go out about every other week to do repeated sampling on the lake at a set of stations. And so we're all there as a team together. It also involves students. So these are Lake and Zuri, who were research experience for undergraduate students a couple of years ago, helping out and developing their projects around questions that are occurring with cyanobacteria on the lake. Eliza, a Midcoast employee who works closely with Patricia, coming back to the lab to process those samples. Patricia, of course, has touched basically like every piece of this workflow through the time that she's been here. And then our current summer student, Sadie, who is working on the Bigelow EGOT or Triple Crown, depending on the reference. Sadie is from the Camden area and started her Bigelow journey as a Keller Bloom student, which is a student for a program for high school juniors from across Maine to come to Bigelow for a week to get immersed in ocean science and aquatic science more broadly. And then she is now a student at Wesleyan and came this fall for our Sea Change semester program. So students in college will come and stay in the residence for a month or for the entire semester, take classes, and work on a research project. So she worked with Rachel and I collaboratively on a project and then came back this summer. So we hope to see Sadie back for grad school or postdoc someday soon so she can just fill up all of those Bigelow accolades. And so the point that I want to make here is that this is kind of the Water Health and Humans way, is involving this whole community in every step of the process so that the folks at Midcoast Conservancy who have that relationship with the people on the lake have been at Bigelow, have done the scientific work. If they're interested, the opportunity is there so that that connection isn't just a report that lands on their desk one day, and they have ownership of that, can ask questions, and can actually help drive some of that research. So like I said, we're on our last summer of fieldwork for this project, but we can already see some interesting patterns. So this is just a plot of time on the bottom here, and then amount of cyanobacteria on the y-axis, the vertical axis from low abundance to high abundance. And so what we see is there's actually 3 main species that we have on Damascotta Lake. We have kind of an early season species that we call this curly morphotype, looks curly. We have Planktothrix, that's that species that kind of brought this whole issue to the attention of everyone just because it's so striking when it is occurring. And that has recurred every year, more or less. Sometimes it's a larger area of the lake, sometimes it's longer duration, but it's occurred every year since then. And then this straight Dolichospermum, another species that we get sort of later in the summer and into the fall. And so when we're out there and synthesizing these data sets, one of the things that we're trying to do is become more predictive and proactive. And so one of the things that we've learned along the way with our handheld meter here that we can lower down into the water, that measures something called phycocyanin, which is a pigment that only cyanobacteria make. And by looking at where that pigment is in the water, we can tell, okay, it's headed towards the surface. We're probably going to get a bloom here in a couple of weeks. And Patricia was just out on the lake today, helping some folks to deploy an experimental buoy that's going to be monitoring phycocyanin, among other things, every 15 minutes across the season. So through that, we're going to hope to get even more predictive. This one, we're only getting that data when we're going out every other week. But with that better resolution, we're really excited to see where that leads. Okay, so we can track which species are there. We can track their abundance. We can start to learn some things about their ecology, but we're also really interested in the toxin production and the potential for toxin for these species because of that direct impact on us. And so the thing about cyanobacteria is that just because you have cyanobacteria, that doesn't necessarily mean that it's going to be producing toxin or that it even has the ability to produce toxin. And so to dig into this a little bit deeper, you could have these 2 examples here, these 2 forms of a species, and one has the code that allows it to make the toxin, and one doesn't have the code. And what we do, so here's this same picture of Sadie, and what she's actually doing in this picture is sequencing the DNA that allows us to look at the code that tells us whether that toxin can be produced or not. And this has really been somewhat revolutionary for us. So this DNA sequencer is about the size of a stapler, and it used to be that we would need to send a sample out to a lab maybe a few states away, and they would be sequencing this on something that took up half of a room, and you'd get your results back in a few months. Now we can collect a sample, we can put it on the sequencer the same day or the next day and start to get results within minutes. So that has been really great for us to really increase our response time and our understanding of these phenomena. So when we do that, what have we learned about the Damariscotta Lake species? All of the different species on Damariscotta Lake lack that code. So they're not making that toxin that we're mostly looking at, which is the toxin microcystin. But we've looked at other toxins as well, and they lack the code for most of the other well-characterized cyanobacteria toxins. So that's good news for Damariscotta Lake. But what happens when we look across the landscape more broadly? So this is a map of kind of our little region of Maine. So Damariscotta Lake there at the bottom with the black arrow. And that pie chart that you see, each sort of segment corresponds to one of these species of cyanobacteria that can produce this toxin. And if the pie chart is empty, it means that toxin is not, it doesn't have the code. It lacks the code for that toxin. If it's filled, it has the code. So Damariscotta Lake lacks the code, so it's all white there at the bottom. If we go a little bit further towards the Augusta area, you have places like Weber Pond and China Lake, where they have all three species, and all three species have the code to make the toxin, which is a really interesting question. An active area of research for us is what gives? Why do some places have these toxic species and some places do not? If you look a little bit further north in the Belgrade area, you can see right in the heart of the Belgrade region, good news for everybody. There, they're not seeing, there's a bunch of those empty white pie charts, so they're not as many toxic forms there, but other things nearby. And just to the west of us, places like Sabatis and Pleasant Pond, you also have these. And so there is this sort of, threat across the landscape, but I also don't want to scare people or shame anyone or make someone upset who does live or have, a close relationship with one of these lakes. Because just because you have the code to make the toxin doesn't necessarily mean that a lot of toxin is being produced. So there's multiple steps here that go from you have a cyanobacteria to you have a very toxic bloom. And we can talk more about that, but that is, like I said, a really active area for us to understand. And the beginning of this is to just understand the scale and the distribution of these different forms so that we can dig deeper and understand this in a more holistic way. And so that brings us to another project right in our own backyard for Adams Pond and Knickerbocker Lake to understand a lot of different water health metrics, which Rachel will talk about in a little bit. But for my part of that collaboration, we've been focusing on, again, on the cyanobacteria and understanding, well, what kind of cyanobacteria do we have right here in our local water drinking supply sites. And the good news there is it's the same species as Damariscotta Lake has, and that it lacks that code. We don't really have the answer yet of why some places have one species and other places have another. And it doesn't necessarily mean that we can just look at this one time in 2023 and not ever go back. So we are collecting samples and working with the water district in an ongoing manner to look at those samples, to triage them, to make sure, okay, it's the same genotype or it's the same form this year as last year. We're going to stay on top of this and keep looking at it. But to actually talk for just a second about what you're seeing here, this is the way that we collect our samples. So that's a filter, that kind of gridded round disc there is a filter. And so what we do in the eDNA work is we take a water sample. You could also take lots of other types of samples, but I'm going to be talking about water samples. And you pour maybe a cup or a cup and a half of water through that filter, onto that filter. It captures, it's a really small, has really tiny pores. And so it's going to capture, this size is going to capture basically bacteria and up. And so the cyanobacteria, these are colonies of cyanobacteria. So they're the green dots. They're actually big enough to see with your eye. But you can also see we've got some sort of bug, some invertebrate there in the center. But there's also a lot of bacteria that you can't see on here with the naked eye. And there's also other algae and organisms and cells of other organisms. And so that's what we call an eDNA sample. And you can do a lot of really valuable things with an eDNA sample. And it also represents an archive of sorts. Once you've extracted that DNA, if you're storing it safely, you can go back to it in 10 years. You can go back to it in 20 years and ask, was this organism I'm interested in present at this location? Was it more abundant? Was it less abundant? All of those sorts of things are possible. So in the first example that I've been talking about, you could look for the toxin genes, the toxin potential. You could also quantify just about any organism to say exactly how many of this organism are in this sample. You can also do something called microbial source tracking, where you can ask where a type of pollution is coming from. So is the pollution human caused? Does it link back to a septic? And is that the restoration activity that matters the most? Or does it link back to dogs and we need to move the dog park farther out of the watershed or put a buffer between the dog park and the lake? So that can be really valuable because we have limited management dollars usually to target them for the biggest bang for the buck. Then the other thing we can do is something called DNA metabarcoding, where you're getting a whole community snapshot. So in this example, all of the fish you could imagine in Knickerbocker Lake or Adams Pond, you could identify and quantify the relative abundance of those fish in a sample. And that's really good for looking at community response. So if you've taken a restoration effort, if you've removed a dam, which Rachel will talk a little bit about, then you could see, did that increase the diversity of fish that we're able to get further upriver and things like that. So those are just some of the examples that the science that we do with our local communities and the things that we're able to bring to bear to put that science to work. And that's where this really, the boots start to get muddy at this point. So these are pictures from Midcoast Conservancy, where they have a youth conservation corps that's doing things like making sure that runoff actually gets to a spot and sent elsewhere instead of straight down into the lake, or some plantings of a buffer near the shoreline. And then this is Patricia, an event that they have called State of the Lake, where we're educating the folks all across the lake on what we're learning along the way. There's other outreach activities that we've been doing as cyanobacteria kind of become more of a hot topic issue in Maine. We formed this Maine cyanobacteria group, which has been really valuable to get feedback from folks across the state who are dealing with similar issues. And we actually recently received a grant to expand some sampling, but also really work on communicating the threats around cyanobacteria. So it's a tricky thing to say, there's cyanobacteria in my lake. Should I let my grandchildren swim in it? Should I let my dog swim? That's a personal risk decision. And so that's not my area of expertise. So kind of convening the folks who need to be in the room, the public health experts, the marketing experts of how do we get the message across, that's been a really valuable process. And so I think with that, we're going to take a bit of a pause for some questions, and then I'm going to hand it back over to Rachel. So that's a great question. It was, what effect does pH have on these different types of bacteria? So pH is one of the metrics that we're monitoring with that that sonde that I was talking about, that meter that we're able to put into the water. And when we do these models that are asking, okay, which of the parameters that we're measuring is predicting or doing the best job of predicting them, pH does not typically come up as one of the more important predictors of them. I think the scale of pH changes in these lakes is more stable than I think the temporal dynamics that these cyanobacteria are encountering. So the question was, what can one learn from the smell of tap water? So I'll give just one quick cyanobacteria answer to that, and then let Rachel. So is this compound called geosmin, I think it's pronounced, that a lot of cyanobacteria can make, and it's called one of these taste and odor compounds. Because if you have these cyanobacteria abundant in a drinking water reservoir, they may not be making a toxin, but they do give the water a distinct taste or odor. And so they are classified as, a nuisance for that reason. So you certainly can tell a lot if your nose knows, right? So if the water smells off, there's likely a reason for it. It can, the geosmin's not going to hurt you. It just doesn't smell great. So that's where, talking to the drinking supply folks, talking to the water health experts is going to help determine, is that smell something that is dangerous, or is that smell something that is more of a nuisance?
Seasonally less appetizing, right? But perfectly safe to drink.
If the cyanobacteria on Damascotta Lake don't have the code and therefore are not harmful, it is still a change. The gunk looks wrong. Do you mitigate it?
Yeah, that's a great question. So I meant to say this in the talk, but there's still a nuisance. We have harmful bloom, we have nuisance bloom. I would definitely categorize, especially that Planktothrix species, as a nuisance bloom, because the way that it looks across the water surface, if folks have been out and seen that, it looks like bright blue snot pieces, and they can be this big. It is not great if you're trying to swim there or boat. So yeah, that is, we are working with Midcoast Conservancy, because we ourselves at Bigelow are not doing those restoration activities, but we are informing Midcoast and trying to give them the best information possible so that they can use the tools and the resources that they have to take some of those actions. And yeah, so like I said, we're also in our final season of working on that. So certainly more to come in the next year as we put the full data set together.
I also would add that I think it's consistency, right? So every lake has kind of perturbations. It's when you get it every year and it just keeps getting worse every year after year that you know that there's a problem. And usually we don't know that it's happening year after year until someone recalls that this has changed, right? There are a lot of strategies that can mitigate things like harmful algal blooms, but the best strategy is prevention and keeping natural lands natural so that you don't get the nutrients going into the system, or to mitigate them, like we talked about septic. You can identify a region of the lake that may be more impacted, that we can kind of prevent those nutrients from going in. That's a really good strategy, is to get it early. There are other strategies like adding chemicals like alum, which basically binds up the phosphorus so that it doesn't have the nutrient it needs to grow. All of those types of really significant strategies should be taken with consideration. It's not a first step. If you have toxin or you have something that's really, really harmful, then maybe that's a step you need to take. But reducing your nutrient loads is going to be your best option early, because when you disturb a system, it's hard to get it back to the original, the way that it was, right? But that's why we try to build resilience, because 1 or 2 years of perturbation, you might be able to still bring it back to that original health because of that resilience that you've built into the ecology of the system. So it's getting back into the hopeful side. So that was a lot of hope. Let me just say, Robin's understanding that just because you see green doesn't mean you have to be really concerned that your health's in danger. The fact that the scientists are working directly with water districts and other groups to be able to give you that information and be encouraged to be able to get that out to community is really impactful. This is just a picture of Robin. I'm always on the camera side. Anybody else that person? But Robin here working with some collaborators from Zebaiag to collect samples and to work in community for their needs. So Water Health and Humans, it's not just Robin and I, although we are a good team. I'm the director of the Water Health and Humans Initiative, but Dr. Christoph Apley, he's part of this project. He's primarily focuses on PFAS and other petroleum chemicals. Peter Countway, who was Robin's postdoc supervisor, he was here a minute ago, but he really has pioneered some of these uses and is really advancing our work from microbial source tracking to know, is it a human pathogen? Like, what's the source, so we can start to address these issues carefully. If it's a human source, maybe it's septic, we can kind of gear those strategies. Robin, of course, has done a really great job explaining his research. Steve Archer runs Bigelow Analytical Services and does a lot of the biotoxin testing for Bigelow. So we have people with different strategies, and also everyone at Bigelow, right? We move people in for the questions at hand. If we need someone that helps with forecasting, we'll call Nick Record. But this is a collaborative unit where we bring in the skill sets that we have here at Bigelow to meet the needs of the communities and the groups that are asking for this help. So as part of this, we're building capacity and restoring our ecosystem. This is work with the Passamaquoddy tribe, with Zebaiag Environmental Department. That's their research department there for the tribe. And so part of this is we connected with this group because they were doing restoration efforts, removing dams from traditional homeland territories to get that water flowing back the way that it was throughout pre-colonialized times. So we're monitoring the return and the health of fish species from these restored rivers. That's kind of what Raman was describing of the barcoding of being able to see which fish are returning. Are there fish like right outside of a culvert or a dam that they're right there ready to go upstream? Just give them a chance, right? Maybe that's the next step. That gives us the ability to invest and prioritize that restoration effort to get the biggest impact as early as you can. Christoph Apley, he's looking at PFAS, so how much potential contamination is making it into the fish. Ben Twining is part of a collaboration that we have recently gotten funded with EPA. So we got a grant funded, I guess last year it finally came through, to work with the Passamaquoddy tribe to assess PFAS, which are forever chemicals, and heavy metals in subsistence fisheries. So things that people are going out and harvesting themselves throughout the water, throughout the system of the Scudic River, which is we call the St. Croix. We got this funding, we were really excited, ready to go, and 2 days before we were supposed to go sample, that funding was rescinded. It was taken back. We petitioned, and under 2 different administrations, we proved that this research is important, and it's vital to the communities of Maine to know what fish are healthy to eat and where we should fish for them. So this is a great opportunity. We got the funding back. This has been like 2 weeks ago. We get to celebrate that we've proven it twice. But with the landscape that is ahead of us, we need to make sure that we continue to support research in a way that those opportunities are still available in the future. Right. So I'll have a rallying cry in a moment, but we also want to build capacity through hands-on training. Like Robin said, with Midcoast, we have research members from the tribal nations coming to Bigelow, once or twice a year, and they're bringing their samples with them. They get to follow those samples that they collected all the way through the scientific process. Because when they go back to community, they get to say exactly how their samples were treated, how they work, how the reactions happen, and they have this working knowledge of the research that we're doing. And that is an enormous opportunity to gain trust in science, because we've lost that a little bit, right? We've somehow disconnected the science that's happening from the people who benefit from it. And that's something we're really trying to work to get back. Keeping our community safe. I won't spend too much time on this because this is what Robin just talked about, but we're monitoring things like harmful cyanobacteria, the human pathogens that are in the water through microbial source tracking, forever chemicals or PFAS, and nutrients in our lake. We're trying to look at the long-term trends to say, the lakes, our drinking water lakes are healthy now. How do we keep them that way? What are the potential threats? And to buy enough time to fix those threats before they become Well, before we have to accept what a change is, right? Let's keep them as clean. That is one thing about Maine that I love, is that we are willing to fight to keep things the pristine way that it is. We all enjoy it, we all love it, and it is great to be on this side of it instead of this, oh my gosh, we have a terribly compromised system, how do we get it back? Is to start with a really healthy system and trying to keep it that way. That is the best place to be. We should all be fighting for those opportunities. This is part of a collaborative with a Clean Drinking Water Initiative here in the Booth Bay region. The Clean Drinking Water Initiative is to raise awareness and to educate about the importance of keeping our water safe, both in the Booth Bay region, but across Maine and across the US, is we need to understand what's in our water and what we need to do to make sure that we're ensuring the health both today and for future generations. We're also designing approaches to capture new and emerging pollutants, right? So this is that innovation side that I'm really excited about. So one thing that my lab's working on is how do we use kelp or seaweeds to do things like capture things like carbon, nutrients, and they're actually kind of good at capturing microplastics too. Their sticky surface of polysaccharides are able to capture some of the microplastics, meaning that we can use them at the end of our storm drains or maybe wastewater systems to be able to capture some of the things that we don't want in our water systems, right? Before they go into the ocean. The ocean is a vast space. How do we keep it contained as it's going in versus dealing with it in a very large reservoir? Putting microbial communities to work by solving environmental challenges. We're also, sometimes you got to look to through the microscope to find the solution. So we're finding microscopic organisms that work together. We've often looked for, one organism, one metabolism, one species, but like a society, bacteria and phytoplankton and microscopic organisms work together to build something greater than themselves, greater than one species. So we're putting them together in these collaborative networks where they're able to share the responsibilities and do things like remove carbon or capture carbon from the atmosphere or trap nutrients as they flow into a system to prevent these harmful algal blooms and eutrophication. It also, by combining these efforts and capturing them, we can recover them and use them for our own benefit later. We don't have to keep making and finding more nutrients. We can reuse them. This recycling method will help society economically and environmentally. And putting Robin's work to use is using the species that we know that don't have those toxin strains as a way to use them to fill a niche, to fill a space that more toxic species would normally take up. Do they have the same environmental requirements that they could replace one another so that we can edge out the toxic strains for the non-toxic strains and buy our lakes some time? And then we're going to build an ocean of opportunity. We're building the blue economy while keeping an eye on ecosystem health and sustainability. All of our solutions, all of our innovations have our goal in mind is to make this a better world for all organisms on this planet, humans, algae, and everything in between. So what I hope that you share with me is a vision for our next generation. By working together, we can protect access, health, and the joy for future generations of these waters. These memories that you were asked to think about when we first started this talk. We want to make sure that 1, 2, 3, 7 generations from now, they get to have those same connections with the beautiful waters of Maine in a positive way. So what can you do to help keep our waters clean? You can shop with pollution in mind. When you have a choice, make the best one you can for the environment and for yourself. Reduce fertilizer use or nutrient runoff. Again, I love a really green flower, but maybe I don't need to add as much, or maybe I could be a little bit more strategic on which ones I choose instead of the bulk bag with everything in it. I can really assess what's needed, or if it is needed. I can maintain my septic system so that it doesn't drain into lakes or, again, through our watershed into our coastal oceans, because all water leads somewhere. I want to preserve our natural areas. That's one thing that the Clean Drinking Water is working with the Land Trust and the Water District as well, is preserving the lands around our drinking water lakes so that, humans are really good at kind of adding things or taking things away from our environment, manipulating it or changing it in a way that maybe isn't as healthy as it could have been. by preserving our lands around our drinking water lakes, or at least or our shorelines. It gives us that space for buffer, and allows nature to do what it does best. We also need to stay informed and stay active. So knowing what's happening in science, what are the cool things we can learn, and what do we need to know is coming down the pike from for cuts or for other issues that science might face. And I want to talk about science. I've asked this in talks recently, but I'm fervently asking you if we can share it with if you can share what you've learned today with one person who is not in this room, then that impact and that footprint doubles. The more people that know that we are working towards solutions that help each and every one of us, the more people that will fight for science in a way that is clear, that you know this connects to you and it connects to your needs. And this is critical. We don't talk about science enough. And then I want you to support science. Bigelow is a global leader in your backyard, and your support has an impact and enables us to continue. And when I say support, I mean you being here today is a form of support. Everyone has a different way to support us. You can talk to your senators, you can talk to your representatives, you can talk to your neighbors about the importance of science and the importance of what we're all trying to do for our community. There are many ways to support Bigelow, and I'm asking you to use your voice and your time, your energy, and any other capacity to help us keep this moving forward.
Hi, I had a quick question on Robin's research with the algal blooms. I was just curious, do you see that the populations stay in the lakes that you find them in, or are they traveling down the watershed? And I'm just thinking about the map you showed with which ones had toxins versus others. And I know Rachel mentioned the potential of bringing other, algal non-toxic ones to try and fight back. So I was just curious if you could talk on that.
Yeah, that's a great question. So these cyanobacteria are so small and at the same time so robust that they can probably get stuck on the foot of a bird, a loon that's flying from one lake to another. So over the past 10,000 years or so, there has to have been the opportunity for these to move around across the landscape. More recently, with the amount of boats that are moving from one place to another, that's only increasing that movement across the landscape. And yet we still have these kind of set areas where you're seeing a lot of them in one place and not as many in another place. So I have to think that the opportunity has been there for them to be in that other space, and that leads me to think that it's some sort of niche, that there's something about Damascotta Lake that works for those species and doesn't work for the toxic species, and vice versa. And maybe it's a competition thing, but yeah, we don't have the full answer yet. But that also brings to mind the just in terms of this watershed, we did sampling downstream in the estuary to see if we could track these cells leaving the lake and coming into, say, Great Salt Bay or further down. And we noticed that, as you'd expect, pretty quickly the ecology and the salinity change is so abrupt there that we're not detecting them very far downstream. The bad news there is that probably also means they're bursting open. So if they had toxins, they would probably burst out at that point. But so far, we have not toxic types. So more good news. Yeah.
Thanks very much to both of you for excellent presentations. I have an interdisciplinary question. I'm working on sea level rise. My focus is quite macro about economic and political issues. As you were talking, I realized I knew absolutely nothing whatsoever about the kinds of microbiological and chemical processes. Is there any generalization you could make about the effects of sea level rise on the kinds of phenomena that you focus on? Or send me in a direction where I can find out?
I mean, An obvious impact is with sea level rise, there's also seawater inundation. So when storm surges or just sea level rise that lead to stronger storm surges, you get pollution or when the seawater comes in farther, it has access to more things to increase pollution going into to any estuary or to the coastal ocean. That's one. There are also organisms it destabilizes. If you have certain species that are not as salt tolerant, that are native species that really keep your shoreline stable, it can have damaging effects to those plants. Just as humans can, it can, we can disrupt it. The sea can disrupt it as well. There's, I mean, if you give me a minute, I can probably come up with more, but those are kind of two. Sea level rise inundating certain low-level lakes, that's another concern. Seawater inundation coming in to subsurface waters that could add salt to different reservoirs, overtoppings, breakthrough with a lot of stormwater, opening spaces. The list is fairly significant, yeah. It's all feedback, right? It's why these interdisciplinary studies are so important, is because if you look at one aspect, you forget about all of the knock-on effects that one leads to another in that way.
What role does wildlife, particularly aquatic wildlife, play in preserving the quality of water or being a detriment to it? I'm thinking particularly of Canada geese. So.
Yeah. So that's one where if you've got too much of anything is probably a problem. And so if you've got excess geese poop that then when it rains is washing in, that represents a problem. Other wildlife, absolutely critical for maintaining that kind of delicate balance that these ecosystems, they need bottom up and they need top down. And so you need all the predators and you need all the prey to be there for everything to be healthy. Yeah.
And it's key to native, right? Because invasive species can kind of disrupt the way things work, right? One tilt out of balance can affect all the organisms and then how they function in that ecosystem.
So on a personal note, when I came here 7.5 years ago, I'm a nutrient biogeochemist. I worked on nutrient issues, eutrophication, all the things that phytoplankton can produce that are bad. And I did it in the Gulf of Mexico and the Chesapeake Bay were two of the main places I worked, which compared to Maine are just, it's a bad scene. So one of my personal goals coming here was I wanted to make sure that Maine's waters did not turn into the Chesapeake Bay. And so this Water Health and Human Initiative has been a dream of mine, and I just am thrilled at what Rachel and all the people she's working with, like Robin, are doing with it. So our sponsor for this is HM Payson, which is one of the gifts that I think Chris Flower has given her. Chris and DZ Flower, their son, works at HM Payson, and I'm guessing this is the start of this relationship, and they support every year the Café size series. So we're very grateful for that. Okay, so we have 2 more talks this summer. The next one is PFAS in Casco Bay, tracking the fate of forever chemicals on Maine's coast. And this is Christoph Apley, who's a senior research scientist here, and Hannah Sterling, who is a research technician. And that will be a great talk. And then the last one is ocean epidemiology, tracking marine diseases to protect fisheries and ocean health. So that's Maya Groner, who's one of our senior research scientists, Melissa Rocker, who's a postdoctoral fellow, and Ren Yoshioka, who has been one of your bartenders, who is our newest research scientist, who came as a postdoc and went through the same horrific process of having to compete for a job internally, and he just did great. So with that, thank you so much for coming. Thank you for caring enough to come out and support and hear about our work. And I hope you have a wonderful evening and a safe drive home.